In this video, we're going to review the questions that were in the recordings for lipid metabolism. So in this first question, I was asking why it's necessary for these fatty acids to be bound by proteins when within cells. And so let's remember that due to the structure of fatty acids, they have that conical van der Waals radius. They tend to actually disrupt the bilayer because they act like detergents. So B is correct. We got to bind those fatty acids with protein because otherwise our cells would kind of break apart, and no one wants that. Then we were talking about incorporation of cholesterol into the center of lipoproteins. And so we needed to esterify that cholesterol in order to convert it from something that's amphiphilic to totally nonpolar. So if we're going to esterify the molecule and get rid of its polarity, we should be targeting the hydroxyl group. Finally, folks, in the last question, I was asking you to think a little bit about the consequences of having a multi-enzyme complex. And so there were two correct answers here. If we can be passing a single metabolite from enzyme to enzyme within a single complex, that substrate is basically just shuttled from enzyme to enzyme. That kind of speeds things up. Additionally, if that substrate never leaves the complex, and it's been shown that the substrate really doesn't leave, it's not going to cross-react or end up going off in weird metabolic pathways. So A and B were correct here. And that's it, folks.